Welcome to our channel Mootechie. In this session, you learn about reproduction in fungi. This includes the mechanisms of vegetative reproduction, fast-track asexual strategies, complex sexual life cycles, the diversity of fungal spores, and their ecological importance. Fungi are fascinating organisms, and understanding how they reproduce is key to grasping their role in nature. Let's begin with an introduction to reproduction in fungi. Fungi possess a versatile reproductive toolkit utilizing vegetative, asexual, and sexual modes to propagate. This means they aren't limited to just one method. They can choose the best strategy for their situation. Their reproductive strategies switch dynamically with environmental conditions to maximize survival and genetic diversity. So if conditions are good, they might clone themselves quickly, but if times are tough, they might mix genes to create tougher offspring. We will explore each mechanism through clear visuals, distinguishing between simple clonal growth and complex spore formation. Now, let's look at the life cycle for an overview of fungal cycles. First, many species alternate phases, N plus N, called a dikaryon, 2N, which is diploid, and N, which is the haploid stage. This alternation is a core concept in fungal biology. Second, so-called perfect fungi have both asexual and sexual stages in their life cycle, while imperfect fungi are known mainly for asexual reproduction. Third, spores disperse via wind, water, or animals and germinate to form new mycelium. These spores are like seeds but for fungi, and they are how they travel to new homes.
The diagram here shows the core stages: spores, germination, mycelium growth, and fruiting body formation. Think of this as the fungal life cycle in a nutshell, starting from a tiny spore and growing into a fully developed body.
Moving on to asexual methods, we start with vegetative reproduction. This is all about growing copies of yourself without making special reproductive cells. First, budding, common in yeasts. A small outgrowth forms on the parent cell, enlarges and detaches to create a new individual. It's like a little bubble pinching off. Second, fragmentation. Hyphae, which are the fungal threads, break into pieces accidentally or naturally, with each fragment regenerating into a new mycelium. If you've ever broken a piece of mold and seen it grow back, that's fragmentation. Third, fission, a simple splitting of a single cell into two genetically identical daughter cells, common in fission yeasts. It's a very straightforward division. All these methods ensure rapid colony expansion without the energy cost of building complex spore formation structures. They are perfect for quickly taking over a new food source. The table here compares three key methods: budding, as seen in yeasts, fragmentation in molds, and fission in fission yeast. The remaining values follow the same pattern and can be referred to in the table. This table helps you visually distinguish these common vegetative processes. Now, let's delve deeper into asexual reproduction in fungi, which often involves making specialized spores. You'll see terms like sporangiospores, conidia, and conidiophores. Sporangiospores are produced inside a sac-like sporangium atop a stalk. Imagine a tiny pouch full of spores. Conidia are formed externally on a specialized hyphae called a conidiophore, which is very common in molds. They often look like chains or clusters of dust. The advantages of asexual reproduction like this are very rapid clonal multiplication and wide dispersal via wind or water. One fungus can produce millions of identical spores in a short time. This occurs typically under favorable conditions, creating massive spore loads in the air around us, which is why molds can appear so suddenly. For genetic diversity, fungi turn to sexual reproduction. This is a more complex three-step dance. First, plasmogamy, the fusion of cytoplasm from two compatible mycelia, resulting in a dikaryotic stage where cells contain two separate nuclei. They've merged their bodies but not their genetic cores yet. Second, karyogamy, the critical step of nuclear fusion, creating a diploid zygote nucleus. Now the genetic material combines. Third, meiosis. Reduction division follows rapidly, restoring the haploid state and producing genetically unique spores. This shuffles the genes. This complex process increases genetic variation and is often triggered by environmental stress or adverse conditions, giving the next generation a better chance of survival. The diagram illustrates this sequence: plasmogamy, followed by karyogamy, then meiosis. It's helpful to classify the types of fungal spores. Zoospores are motile, flagellated spores found in aquatic fungi, like chytrids. They swim through water. Sporangiospores are asexual spores produced endogenously, meaning inside a sac-like sporangium. Conidia are exogenous asexual spores formed in chains or clusters on conidiophores outside the cell. Then we have sexual spores, ascospores which form in a sac called an ascus, and basidiospores which form on a club-shaped structure called a basidium. Both are formed via meiosis. This chart from Mootechie visually groups these spore types, showing zoospores, sporangiospores, conidia, ascospores and basidiospores, helping you see the difference between asexual and sexual spore origins. Now, let's see fungal reproduction in ecosystems and understand why fungal reproduction matters. First, for decomposition. Fungi break down complex organic matter like dead leaves and wood, driving global carbon and nutrient cycling. They are Earth's recyclers. Second, through mycorrhiza. Symbiotic fungi colonize plant roots, significantly boosting plant nutrition and overall soil health. Most plants depend on these hidden fungal partners. Third, in food and biotech. Fungi are essential for producing bread, beer, cheese, antibiotics like penicillin, and industrial enzymes. Fourth, in disease cycles. Fungal and animal plant pathogens regulate populations and shape ecosystem dynamics. Their reproduction spreads these diseases, which can control overpopulated species. These icons on this slide symbolize these key roles: decomposition, nutrient cycling, plant symbiosis, and food production. Finally, we reach the end of our Mootechie presentation. The slide says thank you. We hope this visual guide helped you understand the fascinating world of fungal reproduction. It also encourages you to subscribe and recaps the session. Growth via vegetative means, rapid asexual propagation, complex sexual cycles, diverse spore types, and critical ecological roles. You've now explored the remarkable ways fungi multiply and spread, from simple splitting to intricate spore production, and seen how vital these processes are to our world. Thank you for learning with us today.